Yes, Pavan, let's begin. Okay, message to Jagadgiri Gutta. I think it's a problem with your device because I think everybody else can see us and the video. Pavani, are you there?
Oh my God, I was on mute.
Oh,
I was speaking. I thought I am not on mute.
no
I'm so
worries.
sorry. I'm so sorry. Yeah. So what I was saying is that firstly, I want to apologize to all of you for the inconvenience that happened last time due to the technical issue with our Zoom account, and it's still not fixed. That's why we are having this webinar. Though I'm a little disappointed that I can't see our children and their faces on the screen while the speaker is presenting today, I hope that we have your complete attention throughout the session, and we are very much, we can still take questions from all the students. Only thing you have to do is you have to raise the hand when your student wants to interact with the scientist, and then the host CCMB team can unmute you, and then you can interact with the scientist. Okay. Okay. Not when I said raise your hand, I don't think it's not just ma'am, Krishna ma'am, complete the session. But if it's anything else right now, you just post it in the chat box. But post the session, we'll have an interaction where you can use this mode to interact with the scientist.
Yeah,
And
I would
again,
also say
yes.
the question, there's a question and answer box. So whatever questions you have, if you put in the Q&A box, it's easier to keep it track.
Yes, yes. Please note that there are two boxes. One is chat box, the other is Q&A box. So post your questions in the Q&A box. If you want to directly ask any question to the scientist, please mention it in the Q&A box so that it will be easy for us to identify. Okay, so I welcome again Krishna ma'am. Sorry again for the last time. And then thank you so much for giving us your time. And as I said earlier, Dr. Krishna ma'am is a scientist from Center for Cellular and Molecular Biology. She is a microbiologist where she has done her master's and PhD in microbiology from Osmania University, where she has worked on some strain improvement on optimization studies. And later she worked as a postdoctoral fellow. That means something that you do after your PhD. She worked as a postdoctoral scientist at the Center for DNA Fingerprinting and Diagnostics. And ma'am is an expert in so many molecular biology tools, very cool techniques like PCR, gene knockouts, plasmids. I don't know if most of you would understand all this, but if you are a +2 student or if you're teaching +2, you might have a small chapter on RDNA technology, which covers all these techniques, transformation, what is LAG-C, lacopron, and all that. So ma'am is an expert. She has been working on using all these techniques, most of these techniques for her research work. And she has many awards like the Department of Biotechnology Postdoctoral Fellowship. And then she also qualified in the state government and central government different eligibility examinations for junior research fellowship and also lectureship. But ma'am has a huge research experience of over 17 years in different areas and different industries and research institutions. And we are also glad she also worked as a lecturer for some time, and she would definitely understand. I think she would use that expertise in conveying the message today to the students while walking us through the session and when she was explaining the topic. So it would be easy for our students to understand because ma'am also has some teaching experience, and she has She has numerous publications in national and international journals, and she has presented many papers in national and international conferences so far. So we are very glad that she could join us with all the students today to explain us more about the bacterial cell wall and its importance. So with this few lines, ma'am, I hand over the session to you, and we hope our students will learn many more exciting things about bacterial cell wall during the session, and I hope they'll interact with you after the session.
Yes, there
They
will
have
be
loads of
quizzes done
questions.
at the end.
Yeah, and as Somdata ma'am said, we will have very exciting quiz session at the end of the lecture. So please stay tuned. Over to you, ma'am.
Yeah, thank you, ma'am. Thank you very much for your introduction. And one more thing I would like to say here. So if you are any problem in the students are having any problem in asking in English the questions, they can ask in Telugu. I can answer them, no problem. Language is not a barrier. So now let us start the today's talk. So today I am going to talk about the.
They didn't see.
I have to see the. Take it down. Okay. Hi, students, everyone. Let us start the talk today. So you all know living organisms, yes? Living organisms which are like having growing and then multiplying. So based on their size, you can divide them into two different categories. One is called as macroorganisms, the other is called microorganisms. So the name itself tells you macro. What is macro? Macro means something which is big. That means you can see with your naked eyes. That means without any device or without help or anything, you can see them. So these organisms are examples like plants and animals. When you come to microorganisms, these are micro. Micro means small. They are very small that your eye cannot see directly, but you can see them with the use of a special instrument called as microscope. So microscope, which you can use, you can see the figure of a microscope here. Most of the people might have seen this in your textbooks or on the internet. So then microorganisms, now again they have subdivided into different groups based again on their size. There are some very small, there are some small, bigger than that others. So these are 5 groups. They are called as bacteria, protozoa, fungi, algae, and viruses. So if you see bacteria, what images are you seeing here? The small images you see, there are single cells which are seen under a microscope. So these are like rod shaped bacteria. If you see here, they are round shaped and round, but they are chained. So these are called chain, round and chain like bacteria. And these are like round and like a group of bacteria. So these are the single things that represent the single cell, which you cannot see with your naked eye. But when the single cells grew and form millions of cells form a colony, you can see the colony on the plate. So what you are seeing here is a bacterial colony that has grown in a medium that we call as, this plate we call as a Petri plates. That plate will have a medium for the organism to grow. So when single cell, they multiply and forms a colony, you can see the colony. So this one colony will have approximately 10 to the power 9 cells in it. You understand? 10 to the power 9 cells. Then coming to the protozoa. Protozoa like amoeba, paramecium. So these are the things which you cannot see, but you can see the infections caused by them. So this is infection caused by a protozoa. Like similarly fungi. So most of you're aware that when you leave bread at your home for a long time, you see this kind of growth, right? Green and whitish color kind of thing. So this is nothing but it is a fungi. So that is the mold or a Aspergillus. So if you see them under the microscope, their structure is like this. And if you see here, this is penicillium. Penicillium is the one which produces your penicillin antibiotic. Then coming to the algae. So most of you must have gone to the beaches or to the lakes nearby, right? You can see these greeny things on these rocks and all. So these are all nothing but algae. So they are green in color. Most of the time algae are, majority are green in color. Some of them are red and other colors, but majority of them are green in color because they have chlorophyll. Then coming to the viruses. Viruses are the more tiny organisms. And recently you must have heard about this coronavirus. We're all familiar with this word, right? Corona and COVID. So this is also caused by a virus. So the structure of coronavirus you can see at the bottom. So it is under, this cannot be seen by your eye. So this is by powerful microscopes, they will see these structures. Then if you see this chart here, you can see how the sizes, they vary in sizes. For example, if you see, most of you heard the term atom, right? So atom is the smallest one, which is a 0.1 nanometer. So if you, before going into this, I would like to explain you on a scale here. So you, all of you must have seen normal scale, right? Which you use in your scale, in your classes to draw lines and all. So this is 1 centimeter. From 0 to 1, it is 1 centimeter. And the small division here, we call it as millimeter. So if it's micrometer, means 1 millionth of this size. So it is this 1 millimeter, if you divide 1000 times, so that is the size of a micrometer. So you imagine what is the size of that. So most of the microorganisms are in the micrometer range. So if you see, you can see the increased size range here. So atoms, and then DNA, and then bacteriophages. Bacteriophages are nothing but viruses that attack bacteria. Like you are having viruses which attack humans. Similarly, you have viruses which attack only bacteria. So they are called as bacteriophages. Then you have bacteria, so which are ranged from 0.5 to 5 microns. And then mitochondria, which is one part of it is called a powerhouse of the cell. And then human cell. So this is how the sizes vary. So though they draw big structures here, but you cannot see them with your eye. So then now for the present lecture, it is very important for you to understand the difference between a bacterial cell and an animal cell. So here two cells you can see. One is bacterial cell, the other is animal cell. So if you see, there is a nucleus in the bacteria, but this nucleus is not surrounded by any nuclear membrane. But in eukaryotes or in animal cell, you can see the nucleus that is surrounded by a nuclear membrane. Then you focus mainly on these three things here on bacterial cell. You have a cell wall, you have DNA, and you have ribosomes. So if you see in animal cell, are you seeing the cell wall in animal cell? No. That means animal cell doesn't have any cell wall. Cell wall is very specific to bacteria. Only bacteria will have the cell wall. Then coming to the nucleoid, DNA, the difference, both bacteria and animal cell has the nucleus or DNA. But in animal cell, it is protected by a nuclear membrane. But in bacteria, there is no nuclear membrane. The other difference between these two is ribosomes. Both bacteria and animal has the ribosomes, but the size of ribosome is different in both. So you have to remember these three differences in later slides. It is very useful for you to understand the work. Then these are some of the differences which I spoke. I have given a tabular form so that you can have an idea. So bacterial cells, they are of 0.2 to 2 microns in size, and animal cells 10 to 100 microns. That is more than 10 times more than bacterial cells. And here the cell wall is made up of peptidoglycan in bacteria. But as I said, animal cell doesn't have a cell wall. And bacterial cell doesn't have a membrane bound nucleus, and animal cell have a membrane bound nucleus. They do not contain mitochondria. Animal cell has a mitochondria. And the other difference between bacteria and animal is the ribosomes. So bacteria has 70S, that is a small ribosome, whereas animal cell has a big ribosome, that is 80S ribosome. Then where do these microorganisms live? So they are there everywhere, like soil, water, plants, and animals. Only thing is you cannot see them with your eye, but their presence you can feel either in the form of a disease or in the form of the beneficial effect to your body. Now let us come to our body. So our human body also has many organisms, like in your nose, mouth, stomach, gut, vagina, and skin. There are many microorganisms. Some of them are useful to you and some of them are harmful. If the useful bacteria is there, you are going to benefit from the bacteria. If the harmful bacteria is there, you are going to get a disease. So what are these? What are these good bacteria and bad bacteria? Now coming to good bacteria. So you must have daily, you must be taking a curd or something at your home, right? Perugu or majjiga, you call it in Telugu. So that is one kind of good diet. This is good because it has a good bacteria. So if you see the soya sauce that is done by aspergillus, and this cheese or yogurt which is having this lactobacillus, that is the milk converts into curd with the help of this lactobacillus. Okay, and beer and wine and bread. Even the bread also, only yeast is required for making bread. So these are all good bacteria which will help you in making your food and giving you some strength. So recently we are having this probiotics. All these organisms are probiotics. How these probiotics enter into our body? Okay, you have to consume the food which they contain. For example, daily in our routine work, you use curd, idli, dosa, or pickles you take, right? So all these are called fermented foods. So most of them have these microbacteria or probiotics, we call them, so which are useful to your body. So that is one side of your story. The other side is bad bacteria. Bad bacteria are the one which cause disease. So they are not useful for us. They are going to harm us. So there are some, here I am depicting some of the bad bacteria or disease causing bacteria. So the Staphylococcus aureus, Streptococcus pyogenes, and Streptococcus pneumonia, Bacillus, E. coli, Salmonella. So Salmonella, which is the one which causes you typhoid, Vibrio cholera, which causes cholera disease, and Klebsiella pneumonia, which causes pneumonia. So all these are some of the bad bacteria which will lead or which will cause you a disease. So if you want, you can, there's a table here, you can see bacterial disease caused by the microorganisms. So most of these diseases, if you see in the table, they are caused by bacteria, but I have listed some of the diseases which are caused by viruses also, like your influenza virus and pulmonary virus, and then hepatitis. So chicken pox. So most of the people, you must have known this experience of chicken pox when you are young, and then common cold. So all these are like viral infections. They are caused due to virus. And dengue fever. Recently there was a outbreak of this dengue fever, right? Then the next question comes, the good bacteria we don't have to worry about, right? But we have to worry about the bad bacteria. So how to control or how to kill these bad bacteria or disease causing microorganisms? So the answer for this is by using antibiotics. So what do you mean by antibiotics? So antibiotics are nothing but which you use against living organisms. So anti means against, living, bio means living organisms. So what do these bacteria antibiotics will do? They kill or inhibit the growth of bacteria so that there is no spreading of bacteria in your body. And basically these are produced by living organisms like fungi and bacteria. And sometimes you can also synthesize them in the lab, but they are semi-synthetic. You should have some biological compound, you can modify it. Okay, then they are different from vaccines. Most of the people think that antibiotics and vaccines are the same, but not. So antibiotics are the ones which you take once you get a disease or your infection. To kill that organism, you take antibiotic. Vaccines are not like that. Vaccines you take to prevent the occurrence of a disease. For example, recently we're all taking these COVID vaccines, right? You must have heard the COVID vaccines. So what is this COVID vaccine? COVID vaccine we are taking so that we will not infect with the coronavirus. So this is the difference between antibiotics and a vaccine. Similarly, polio vaccine, chickenpox vaccine. So all these are vaccines which you take to prevent the disease. Antibiotics are the ones which you take to get rid of the infection once you get it. And if you see here, so this is a Petri plate which I have shown in the first thing. This is all the bacteria that is growing on the entire plate. But when I have an antibiotic in the plate, this is an antibiotic containing paper here. So this antibiotic diffuses into the medium. So wherever the antibiotic is diffused, you can see there is no growth. So this is called as clearance zone. So this is the clearance zone. That means this antibiotic present on this is killing the bacteria that are nearby. So where there is no antibiotic, the bacteria are growing. So these are some of the antibiotics: amoxicillin and clindamycin, doxycycline, erythromycin. Penicillin G is the most noted one. Then what is the advantage of these antibiotics? Why should we take the antibiotics? Okay, they are very highly effective. That means they will target the bacteria and kill the bacteria. And the cost of these antibiotics is very low. And sometimes some infections, if the preliminary stage of infection is there, you can cure it by antibiotics. Thereby you can prevent a surgery. And then sometimes after surgery, when you have an operation or something, so post surgery means after the operation, to get rid of this infection and all, you will take the antibiotics. It prevents the disease progression. And the one major advantage of this antibiotics is they are selectively toxic. What do you mean by selectively toxic? They are toxic only to bacteria, not to your body. So they kill specifically bacteria. They don't kill your cells. That is called a selective toxicity. They are targeted to a specific part of a microorganisms. They are not going to harm to your body or your cells. And always there are two sides of anything, right? One use and the other is disadvantage. So here antibiotics are also having the disadvantage. Here the resistance development. Means if you use too many antibiotics, the bacteria will overcome these antibiotics. They can grow in the presence of antibiotics. So that is called resistance development. And some people will have this allergic reactions to these antibiotics. That is a very minor thing. If you are allergic to one antibiotic, you can shift to other antibiotic. So these are the things why again I'm iterating here the differences between bacteria and animal. The three major differences I have narrowed down here. Only three major differences I am talking here. One is cell wall, the other is nucleus, and the third is ribosomes. You keep these 3 points in mind. So now antibiotics, how these antibiotics can be taken. So if you see here, antibiotics either you can take as a tablets or you can take as a injections. So you can, these are the tablets and these are the injections. So depending on your requirement, if the infection is mild, it can be controlled by tablets. But if you are having a severe infection, you need high dosages of antibiotics, then you take injections. Then coming to the mechanism of antibiotics, how antibiotics are going to kill the bacteria. So now here the previous slide information will be useful to you. So one is inhibit the cell wall synthesis. You can see here the cell wall is where cell wall is present. It is present only in bacteria, right? So the antibiotics will act only on bacteria. So there are some common antibiotics which can be used here: penicillins, cephalosporins, and vancomycin, etc. Similarly, the nucleic acid synthesis. So I have told you that nucleus is different between bacteria and animal cell. So here in bacteria, nucleus is directly exposed. There is no nuclear membrane. So there are some antibiotics which specifically affect the DNA of bacteria. And the other thing is protein synthesis. Protein synthesis where your ribosomes come into picture. So I told you that ribosomes are small in bacteria and large in animal. So these antibiotics like macrolides, clindamycins, all these things, they inhibit the 50A subunit, and the other thing, they inhibit the 30A subunits. So directly they don't allow the protein to synthesize. So if you don't have a protein synthesis in your body, bacteria will not have the energy, so they will die ultimately. The next problem I told you that this antibiotics, there is a disadvantage. There is a resistance comes when you use more antibiotics. So how this resistance happens? So when you're having lots of germs in your body, let us say, and you have taken the antibiotic, so all the cells are dying. So these translucent things, they are dying. But one is very stubborn, like in your classroom, when a teacher comes, everybody will keep quiet, right? But one or two students are like that, they may keep on doing the noise in the room. Similarly, here, this single bacteria, it is stubborn, so it can resist the bacteria antibiotic. So this starts multiplying. So you see, after some time, the one bacteria have multiplied into many. So this is how the antibiotic resistance comes. Similarly, the bacteria will also have its own defense mechanisms, right? So when somebody wants to hit you, you defend yourself, right? Similarly, bacteria also will defend. How they try to push out the antibiotic from inside to outside. Antibiotic is entering, but there are some efflux pumps which they will pump the antibiotic out, so there is no effect of the antibiotic. And some other mechanisms where they change the target so that it cannot bind. And other thing, that modify the target, they increase the target sites. You can see initially there is only one target site, but when it sees antibiotic, it will produces more targets. So now the antibiotic what you have taken is not sufficient to kill. Similarly, it modifies the structure of this antibiotic target. Now the antibiotic cannot bind. Then why this antibiotic resistance is coming? So here is your public knowledge or your public awareness is very important here. Okay. So when you go to doctor for any small thing also, some doctors, they will write antibiotics, but some doctors, they will say, you don't require antibiotics, take rest. So that is one. So over prescribing of antibiotics, when it is not required, you should not use antibiotics. For some disease conditions, you don't require antibiotics. For viral diseases, you don't require antibiotics. Just to avoid secondary bacterial infections, you take antibiotics. And you should use only that many days. If doctor says for 5 days, use it only for 5 days. Don't use it for 10 days. Similarly, patient, that is one. Then unnecessary antibiotics used in agriculture. Nowadays, what is happening? All the animals like hens, goats, fish, all cows. So they are using these antibiotics to prevent infection in animals. But finally, we are eating that material, right? So antibiotic is entering into our body. If it is in a small quantity, then we don't have any problem. But nowadays, people are using more antibiotics in these areas. So indirectly, we are consuming more antibiotic. Then poor infection control in hospitals. Okay. Then what do we do in research labs? Now, so far what I have told you, I have explained you what is bacteria and how it is different from human cell, and what are antibiotics, and what is the use of antibiotics, and what is the problem with the antibiotics. So the major problem here is antibiotic resistance or superbugs. That means the organisms which overcome these antibiotics and they will grow. Like Superman, they are superbugs. So our research area, research labs, so most concerned about this problem. So it is more dangerous than your COVID-19, this antibiotic resistance. So that's why you should, whatever antibiotics are there now, so most of the organisms are developing or developed resistance to the antibiotics. So now what is the requirement? We need to discover new antibiotics and identify new targets. And so for doing these two things, research comes to rescue. How to identify new antibiotics or how to identify new targets for antibiotics. Now again, just to recapitulate, I showed you here the bacteria. So this is the bacteria Escherichia coli. Normally we call it as E. coli, so which is a rod-shaped bacteria. So we all were in the lab, we all use this organism as a model organism. Now coming to the cell wall. Basically, our lab is interested in understanding the cell wall of bacteria. So if you see here, so bacterial cell wall is made up of 3 layers. There is an outer lipid layer and a peptidoglycan layer and a plasma membrane. So we are majorly interested in understanding this peptidoglycan because most of the antibiotics are targeted to this peptidoglycan. So if you want to see the analogy of your bacterial cell to your school, you see cell wall is like your fence. So anybody enters into the, nobody can enter crossing this cell wall, right? You have a gate, which is like a cell membrane. So only the people who can permit, they can come. Everybody cannot come. There is a selective entrance here. So like this, if you see nucleus is like your principal office and your nucleus is like this, you can see all the cell you can put as a similar, compare with your school, like ribosomes, like your classrooms, which provide you the knowledge. Now, what is the importance of cell wall? So there are two aspects of the cell wall. One is what is the importance of cell wall to bacteria, and what is the importance of cell wall to us or to humans. So for bacteria, it is mainly to retain the shape of bacteria. So if the cell wall is not there, bacteria will not have its shape. The rod-shaped bacteria, if they don't have cell wall, they become round. And similarly, they should resist internal turgor pressure because inside there is cytoplasm. So the cytoplasm cells in a dynamic motion and it is moving, it is increasing in size. So in order to maintain that, there should be a strong cell wall. And also the pathogenicity should not enter other microorganisms infected by other things. For research, what is the use? So mainly peptidoglycan we use for our studies. And what features we are interested in? We are interested in to study how bacterial cell wall is made. Means once you know how it is made, then you can think of destructing it, right? And then identify important proteins that require for the cell wall preparation and search for new drug targets and selective drug target. So this is the molecular structure of your peptidoglycan. So if you see the peptidoglycan, so it is chemically made up of sugar molecules. I think at least the 10th class students are aware of this sugar molecule structure. So these are two different sugars, N-acetyl glucosamine, this is called, and this is called N-acetyl muramic acid. And this N-acetyl muramic acid, it is connected to 4 circles, you can see, right? They are called amino acids. And 2 amino acids of adjacent chains are linked by a cross link. So if you don't have cross link, the cell wall collapses. And what other things you should know here? So basically, if you look at here, so there is a peptidoglycan is having a glycan chain, 2 glycan chains, and having a peptide. And these peptides are cross-linked so that there is a net-like work around the bacteria, which gives protection. Once this network is disrupted, the bacteria cannot survive. So then fundamental question in microbiology is how bacteria grow and divide while maintaining their cell shape. So this is the major question. So most of you are aware how bacteria divides. Okay, so for here I am depicting the same thing. This is a single cell. So as the cell grows, you can see there is an elongation. The cell wall is increased. The nucleus is divided into 2. And the nucleus moved in the DNA or chromosome moved into 2 different positions. And there is a cell wall constriction and there are 2 different cells. So what you have observed here, finally, you got 2 cells out of 1, but both of them are having the same size, same shape as your parent, right? So how is this possible? The major question is here. So you're having, you can see here, the cell is small in size and it is elongating. So to elongate this way, your peptidoglycan should also elongate, right? Your cell wall should elongate. How is this possible? Just imagine a wall. So how do you increase the length of a wall without breaking it? It is not possible, right? If you want to expand the length of a wall, either you should construct at the outer surfaces or you should break in between and attach new bricks so that you can increase the size. So this is what happens. You can see here, this is a normal resting cell. When it is dividing, you can see there is an enlargement here. So this enlargement happens. See there, I have already shown you there's a cross link between 2 peptides, right? So this peptide is cut. the cross-link is cut by an enzyme. These scissors indicate an enzyme. So there is a place. The two strands are separated, right? You can see here, the two strands are separated. And the newly synthesized, the pink one, comes into the picture, and then it forms cross bridges again. Like just you imagine, you all people are standing in a circle, holding hands in hands. If one of your one new person wants to come and joining into your circle, so what you should do? One of you should break your link, and you should allow the new person to come in, and then you can form a new link with the new person. Again, the circle is complete, but now the circle is bigger because one person has extra come. So now it is clear. So like that, the bacterial cell also elongates. So this is the depiction, the same thing I have shown here. So there is a cross-link cleavage. So in the lab, whatever we are working, we have identified these cutter proteins. So what are the proteins that are cutting these bonds between two adjacent things? And there is an addition of new peptidoglycan, the green color, and there is an again cross-link formation. So that means basically you are having enzymes which synthesize and which cut and again which form the link, right? So these penicillins, where they will act. So penicillins majorly they will act on the synthesis. You have this penicillin binding. This is the penicillin antibiotic. So this, the pink color you can see here, this is the penicillin. So this is one of the enzyme which forms a cross-link between the two. But when penicillin is there, this cannot bind here. So there is no cell wall synthesis. That's how the bacteria dies. So what our lab has done, so by using all the techniques which your madam has described, so we're using all those techniques, we identified in the lab 4 different enzymes. We call it as MAPH. We give some names to the enzymes, like how you are having your names. So the enzymes also we give some names. So MAPH, MAPS, and MAPM, and MAPK. So these are the 4 cutter enzymes which cut the peptide bonds between the two pentapeptides, and they give a space for the newly formed glycan chains. So then what is the use of this research? You might be thinking, why we are understanding the cell wall structure, why we are identifying these cutter proteins, what is the use? So once you know these are the cutter proteins, so if you can design an antibiotic which can bind to this cutter protein. Remember, these proteins are there only in bacteria, right? So if you can develop a antibiotic which can bind to this cutter protein, now the cutter protein cannot act. So there is no breakage of the cross link, and there is no incorporation of the new material. Finally, the elongation of the bacterial cell stops. So that's why it does. So these, our idea is to use these cutter proteins as a new targets for antibiotics. So thereby you can have new antibiotics, thereby we can overcome the resistance to existing antibiotics. Okay, thank you very much for listening. And if you are having any questions, you can ask in Telugu or English or Hindi. You are welcome.
You can raise your hand. And if you raise your hand, I can unmute you and you can ask the question directly. Yes. So TSWREI, I don't know which name. Yeah. We cannot hear you.
Yeah, ma'am. TSWREI. Could you please? Please mute the device, ma'am. It's very disturbing. Please mute it. And post your question in the Q&A box. Yeah, I think we can hear you now. TSWREI. Can you unmute again?
Okay, in the meantime, there's a question from Jagadgiri Gutta in chat box. Are there any alternatives for antibiotics to cure the diseases?
So far, there is no alternatives for these antibiotics. You have to use antibiotics, but only when it is required.
The question, what are the self-antigens?
Self-antigens are nothing but these antigens are different from antibiotics. Okay, self-antigens are the ones which are against our body. Self-antigens will come in the immune response, not in the antibiotics.
Ma'am, from the question, I feel maybe you can speak one or two lines on what is antibody and an antibiotic. I think the students might are getting a little
So antibody,
antibody.
okay, I will tell you. Whenever a foreign body enters into your body, okay, let us say, for example, I will give you the explanation with a COVID virus because everybody knows nowadays. Let us say coronavirus enters into your body. So your body recognizes that a foreign body has come into your picture. Then it raises some antibodies. So body itself will raise the antibodies against the virus, and it will goes and binds to the virus and it inactivates the virus. So that is the antibody. So antibody is produced in your body against any foreign object that enters into your body. Okay. So likewise, we will give vaccines. So what are the vaccines? Vaccines are killed viruses. So when you give the killed virus, the body develops antibodies. Okay. But when you, second time when you are attacked by a real virus, now it has a memory. So the body has a memory. I will tell you with a simple example. Okay. Chicken pox. So most of the children have chicken pox in the childhood. But after they grow, they didn't get chicken pox. Why? Because the body raises antibodies against the chicken pox and it remembers it. So when it has a second time infection, it produces antibodies very rapidly and it kills the virus. So that is antibody. So antibody is our own defense mechanism within the body. So that's why people say immunity. So you should have immunity, means you should have enough power to produce these antibodies. These antibodies are nothing but protein molecules. So that's why you should have good food to keep your body healthy and strong. And antibiotics are the one which are synthesized, which are taking from outside, and they'll also kill the bacteria, but they are not from your body. You are taking from outside. They are synthesized by some other organism and you are taking it.
So there are 4 questions in the question answer box.
Yeah. So the question from TSWREI, is there any effect of antibiotic in human beings? Which I told in my talk, the antibiotics generally most of them are useful, but only the thing is some people may have this allergies to some antibiotics, and over usage of antibiotics will lead to antibiotic resistance. That means
bacteria, not humans.
No, antibiotics will lead to bacterial resistance. So you cannot be, it is not useful for you to take antibiotics more. Okay. The next question I can see from Bhagya Lakshmi, how to kill diseases caused by microorganisms. So this is by antibiotics. First thing is any disease, anything, first your body immune system will come into action. Once your immune system becomes weak, then bacteria will invade into your organisms. At that time, bacterial population is more, so you should take antibiotics to kill the bacteria. So first thing is your body defense, that is by producing antibodies. Second is by antibiotics.
Repeat the question because people are not able to.
Yeah, that's what I'm asking. Okay. The question, Bhagya Lakshmi, is how did bacteria grow and where can it resist? Bacteria generally grow. I didn't get your question exactly. See, like how human beings grow by taking food, bacteria grow by taking food material from our body, if it is in the body. If it is outside, it will depend on other organisms, and it will grow by cell division. That's what I have shown. One cell becomes 2, 2 become 4, 4 becomes 8. That's how the bacteria grows. How, which one? That's what I told you. This I have answered. Now, then next question is, does any disease causes from antibiotics in human beings? So far, no. No disease by antibiotics. The next question, Elaveni Uma, for fungal and viral infections, can we use antibiotics, ma'am? Sure. For fungal infections, you can use antibiotics. Even for viral infections, we use antibiotics, but they will not kill viruses. But when virus infection is there, our body becomes weak. So any bacterial infection can spread very fast. So to prevent this secondary bacterial infections, we use antibiotics. And there are some antiviral drugs available for specific viral infections. But most of the time, viral infections they can cure itself within 1 week or so, but some viral infections are very tough. So for that, you have antiviral drugs. So antibiotics will not act against viral infections. But fungus, you have some antibiotics. Okay. Next from Salim Abi Sheikh. The question is, what is peptidoglycan, ma'am? So peptidoglycan, the name itself indicates, right? It is a peptidoglycan. Peptide means proteins, glycan means sugars. So a molecule which is made up of both sugars and peptides, that is called peptidoglycan. This is in the outer bit, in between two inner membrane and outer membrane of bacteria. We have this single layer of peptidoglycan. Okay, Sindhura, how much time is required to cell division? So this depends organism to organism. Okay, like simple bacteria, I will tell, the one which we are working, it takes 20 minutes to divide. So just imagine, in 20 minutes, one cell becomes 2, 2 becomes 4, like that. Then Lavanya, TGT, can we cure all diseases with antibiotics? We can cure most of the diseases which are caused by bacteria and fungi with antibiotics. The next question is Vasu. Can a single antibiotic cure many diseases? If it's so, why there are many types of antibiotics? The question here is, single antibiotic cannot cure all because the diseases caused by different microorganisms. So it is not the same microbe that causes all the diseases. But there are group of antibiotics. Like I told you, there are cell wall inhibitors, protein synthesis inhibitors, and then the DNA inhibitors. So you cannot use same single antibiotic to cure many diseases because the organisms are different. So that's why you should, and the other thing is the organisms also developing resistance. So, and some people are allergic to one antibiotic, but not to others. So that's why you need many types of antibiotics. Next is Zarina. If we get diseases from, the question is, if you get disease from microorganisms, what are the effects caused by microorganisms? So it's like some thief enters into your body, into your house. Okay, like what does a thief does? It takes away everything of your belongings and goes. Like that, microorganisms, they start eating your body. So finally, you become weak. And if you are not taking proper medication and cure, ultimately the person will die. So that's why it starts growing in our body by using our body cells and all. So it makes you weak. And finally, if you are not taking any care, the person may die depending on the type of infection, type of microorganism. It is not true for all. Okay. Which animal cell is like a human being cell? All humans are animals. Okay. So our cell and animal cells look the same.
There might be some
There might
differences.
no differences wise in the organization wise.
Yeah.
But basic unit,
The basic unit is
the same. Only we are having the organs. That's all. Yeah. The next question, Bhagyalakshmi, is bacteria help to human body? If yes, how? Yes. I have shown some probiotics, right? So these probiotics are the ones which help human body. So these probiotics are nothing but bacteria, the lactobacillus, bifidobacteria, which I have shown in initial slides. So some of the time our body is not able to synthesize some vitamins. Okay, these microorganisms will give you the required vitamins. Generally, the curd and the buttermilk what you take have lactobacillus. They help in digestion. So this is the way bacteria help human body. And nowadays, this probiotics is a major field of research. Next question is bactericidal antibiotics kill the bacteria? Yes, the name itself says bactericidal. Cidal means killing. So bactericidal antibiotics generally kill the bacteria. There is another group of antibiotics which are called bacteriostatic. They don't kill, they stop the division, but they don't kill. Only they stop the division, they stop the progression of the bacteria population. Next question is, Zarina, is there any microorganism which we can see with our naked eyes? So far, there is no micro except some algae which you can see, but there is no bacteria which you can see with your naked eye. Next question, Vasu, does excess use of probiotics affect your health? Definitely anything excess is not good. Okay, you should take the required amount. The same thing in the probiotics.
All right, looks like we are done with the questions that are there. How can we avoid multiple trips?
I think the questions are coming up because it's such an interesting topic, right? Antibiotics and microbes. Very interesting topic. So we have lots of questions.
How can we avoid?
We can avoid multiple drug resistance. Okay, this multiple drug resistance generally you see in tuberculosis. Example, I will tell you about the tuberculosis infection. Okay, tuberculosis, the TB bacillus is very tough actually. So that's why people generally treat the TB with 2-3 types of antibiotics. So that bacteria, and they will give for a prolonged periods. You should use for 9 months. Definitely you should use 9 months the antibiotics. So the multiple drug resistance can be avoided only by taking the prescribed amount and prescribed period. If they say for 9 months, you should use for 9 months, though you are not having any symptoms. For example, any fever also, if they say for any fever, if they prescribe for 5 days, you should take all 5 days. Once you many times, the symptoms goes in first, second days only, but you should not stop the antibiotics. Why? Because there will be 1 or 2 bacteria that is hiding somewhere, so that will develop drug resistance. That's why you should use the use of antibiotics in the prescribed way and prescribed format will avoid this multiple drug resistance. Okay, next question is, any virus in our human cells? The virus is there in our cells, we cannot live. Okay, so I think there is no useful viruses like that. So far, we come across only useful bacteria.
Antibiotics is a chronic diseases caused by
Is there any chronic diseases caused by? Okay, next is Zarina. Is there any chronic diseases which are caused by microbes? There are tuberculosis, leprae, all these are diseases caused by microorganisms, right? They are all caused by bacteria.
By chronic, I think they're talking about diabetes or spondylitis.
No. Chronic means it's very severe.
I mean, I don't know what was Zarina thinking
about. You're thinking like it's a very severe disease, right? There is no lifestyle diseases that are caused by microorganisms like diabetes, BP, blood pressure. So these things are not caused by microorganisms. Next question, Venkatesh.
Yeah.
Is
But
any
in
chance
general,
to
chronic is a long-term disease. Yeah, chronic is a long-term disease.
long-term disease? That's what, like
Yeah.
tuberculosis and leper,
Yeah.
and all these are caused by
Liver cirrhosis.
liver
Yeah.
cirrhosis. So many things.
Yeah.
Okay. Then next is Venkatesh. Is any chance to use antibodies to treat if a patient is not able to produce it? Yeah, this is possible. This has been tried for this COVID also. But how these the antibodies cannot remain long in your body. The antibodies produced by your own antibody, there's a continuous supply. But the antibodies which you are taking from outside, they have their own shelf life. So means you should be taking a course of antibodies. This Kavita, what is the question? I didn't understand. Is it require more time for cell division? The time required for cell division in a normal conditions is 20 minutes for the bacteria which we are working. So again, you should remember each bacteria will have their own cell division time. Some bacteria will take long time to divide. Some bacteria divides in 20 minutes. Some bacteria takes 1 day. Some bacteria divide in 20 minutes, 1 hour, like that. So normal E. coli divides in 20 minutes, whereas tuberculous bacillus, it takes more time to divide. Next question is there only antibiotics to cure the diseases which caused by microorganisms? So far, yes. To cure the disease once you get, then antibiotics is the only solution. But you can prevent the disease from getting by using vaccines. But vaccines are not available for large diseases.
I think the question is more like, are antibiotics able to cure only microorganism causing, diseases caused by microorganisms?
Most of the diseases are caused by microbes only, right? What other diseases you will get?
Well, India is a capital of lifestyle diseases, genetic diseases.
Yeah. Okay. Can we then, Prasad, can we make artificial antibodies against a specific antigen, madam? Yes, you can make by using animals or by using cell culture methods. You can produce antibodies to a specific antigen. These antibodies are very specific. So antibodies raised against one virus or one bacteria are useful only to against that particular bacteria or virus. You cannot use antibodies produced for one bacteria to treat for other bacteria. Okay, Jarina, maybe will microorganisms prepare their own food or they depend on others? So there are some microorganisms like algae, like they are photosynthetic, so they prepare their own food, but other bacteria, they depend on others, like fungi, they cannot make their own food. Even bacteria, they cannot make their own food from anywhere. They feed on something. Either they feed on animals or they feed on plants. Okay, plant pathogens feed on plants, animal pathogens will feed on animals. And in the lab, we grow the bacteria by giving them a ready-made food. So we make some food and then that food they grow. So definitely bacteria need. Yes. They cannot synthesize. Some bacteria are there which are called as photosynthetic bacteria. They can make their own food.
Yes, yes. Zarina, ma'am, if you can remember the classification that we teach the students, the photoautotrophs, chemoautotrophs,
yeah.
Heterotrophs. Yeah, so the autotrophs can make their own food. In that also you have photoautotrophs and chemoautotrophs. And heterotrophs don't make their own food. And E. coli is an example of a heterotroph. So we give them food in the media or the plate that we grow them.
Next question.
They want to know about monoclonal antibodies, ma'am.
So what are monoclonal? Next question is, what are monoclonal antibodies? So monoclonal antibodies means they are produced using a single clone. They are very specific to the particular antigen. So they cannot interact with any other antigen. They are very specific to a particular antigen. They are called monoclonal antibodies. Next question is, what will happen if organisms are not present in our body? This is a good question. Some of these microorganisms, the symbiotic microorganisms which are present in your body, like in each and every part you are having a good bacteria which will maintain the pH of that particular area, and they will not allow pathogenic organisms to come and colonize. Okay, like it is a watchman to your house. So if watchman is not there, anybody can enter into your body, right? Into your house, right? Similarly, if your normal flora, we call that as the microorganisms that are present in our body, we call it as normal flora. That is a naturally occurring microorganisms in the body. So they will give us the vitamins which we cannot synthesize, and they also not allow pathogenic organisms to colonize, and they will give some important materials, or they will help us in digestion. So you cannot survive without this normal flora. So you should have this normal flora.
All right, seems like we are done with the questions.
Any more questions you can ask?
I think today has been the record number of questions of 31.
True. As I said, it's a very interesting topic, and many of us use antibiotics, and they have been, I think we should thank COVID. Many of us got familiar with what is antibody, what is antibody treatment, plasma treatment, and all that.
Absolutely. All right, so then if we are done with the questions, let us do a quick quiz. Yeah. And before I move on to the quiz, let me thank Dr. Krishna Leela so much for the
Thank you, ma'am. Thank you on behalf of TSWREI for answering all the questions so patiently. Thank you so much, ma'am.
Students can contact me anytime if they want any queries, questions, no problem.
Oh yeah, actually I should also tell this on this forum. So CCMB now has a program called as Shadow a Scientist. And in that program, individual students, so single students, they can come, I mean, basically they need to write to me if they're interested in visiting any lab at CCMB. So what you need to do is go through the website, CCMB website, and see what lab you are interested in and why are you interested in it. So send me that short write-up and we can allow you to come to our lab for a day. So if you want to come spend some time with Dr. Krishna Leela, that is totally possible.
Thank you so much. It's a very, it's a very interesting program, Somdata. I really like the programs that you've been doing at CCMB. So again, to explain our science teachers who have joined here, ma'am, what you have to do is please make the scientist, make the students go to the CCMB website. Let them go through the labs. Already through these MIlo CCMB projects, they've been exposed to different scientists and the kind of work that they've been doing, right? Let's say one student is interested in the first lecture or second lecture that we had by Karthik, or maybe later we had a lecture on Deccan ecosystem, and we had a lecture by Tangaraj sir. So they can recollect all these lectures, go back, visit their lab pages, and find out one scientist that they're particularly interested in knowing more about the kind of work that they're doing. Please encourage them, give them some time or guide them how to go through the website, where to find the scientist pages, let them understand the basics of the work that they're doing, and help them prepare the short write-up that Dr. Sundata is talking about. Very small write-up, it need not be scientific. It has to be curious, like Dr. Sundata should know why a student is curious to learn about that particular topic. It doesn't mean that the student has to have complete knowledge on antibiotic resistance if he or she decides to meet Krishna Leela ma'am. Only thing is why they want to learn more about antibiotic resistance. What are they looking forward when they come to visit the lab? What are they looking forward from the interaction? What are their plans? What they want to talk to the scientist or ask them about? So all that things, that 4 or 5 bullets, if they can expand on those bullet points, I'll be very, very happy to coordinate and attach them to Somdata so that I'll help them screen the applications or process it, and then I can coordinate with the principals and the head office to facilitate the visit to CCMB for that selected students. But please, it's a wonderful opportunity. None of us had it during our childhood or the schooling days. Please make use of this opportunity.
And yeah, I mean, like Pavani very correctly said, that the reason we want you to tell us a little bit more about why you want to come at CCMB is because it's such a short duration, right? You come only for a day, couple of hours of that day. So we want you to come a little bit aware of what you are trying to get from that session. That's all. The reason is that we want you to tell us what you want to learn in that one day. So it's not a test or anything. It's only for us to be prepared better. Okay.
Have already participants in the quiz?
Yes, there are 19 participants at the moment, and there are 54 participants in the Zoom. So
okay, they don't want to take the quiz. Why?
Oh, so in the chat
Right.
box, there's a link. So click on it. Come here. It's fun.
It's very much fun, ma'am. So that am I allowed to participate because I'm
Absolutely.
going to win.
Go ahead.
I'm
Bobby.
going to win. I'm an immunologist.
Yeah, you can participate.
Very good.
let
I
me
see a lot of
check.
people suddenly joining. Awesome.
Okay. You want to beat me? I want to beat everyone.
I think nobody can beat you, ma'am. I think you will.
Let's see, ma'am. I've been away from classroom for quite a long time. I might have forgotten the concept.
Nothing you will forget. Once you see the question, answer will come.
Quickly, we are at 50%.
Maybe
like 50% people.
Somnata, maybe we are not motivating them with the prize. Maybe we should include that. Then we'll have all the participants.
Yeah. So today's session was actually the second last session. So after this, we will have only one more session with the scientist. And after that, there will be a proper quiz, a quiz with prize and everything.
Yeah, that would be great. Covering all the sessions.
Yeah, all the sessions
so far.
Yeah.
It's nice. It's nice. Okay, I have joined the quiz, by the way.
Okay, all right. So there are 30 people now. So maybe I'll start the quiz.
Yes.
Okay. Okay, not only you're supposed to give the right answer, but also the faster people will get more points. How does a bacterium cell wall grow? By cutting its cell wall, by stitching more content into the cell wall, both of the above or none of the above? Oh, it's both of the above. You cut the cell wall and then you stitch more stuff into it. So you need to do both. All right. So let's see who's. Okay, let's go to the next question. Oh, the leaderboard. So Raju is leading at the moment, but TSWREI Kodangal is just a little bit behind. Okay, let's go to the next question now. Drugs like penicillin, azithromycin are examples of what? Antibiotics, antivirals, antifungals, or all of them? Yay, most of you got it correct. These are all names of antibiotics and probably things that we all take quite regularly. So we should keep an eye on, that we are taking them only when a doctor is prescribing them, not self-medicate. Okay, let's say who is leading the board now. Okay, the toppers have kept their positions. All right, let's go to the third question. Antibiotics should be taken for all diseases, diabetes, viral infections, bacterial infections. Bacterial infections, antibiotics are not for all diseases. Remember? Let's look at the leaderboard again. Who's leading this time? Oh, TSWREI Society has caught up. All right. We are on question number 4. What happens to bacteria in presence of excess antibiotics? They die, antibiotics don't work on them anymore. They become antibiotic resistant or become superbugs or all of them. All of these things happen, right? Like antibiotics kill bacteria, so of course bacteria will die. But if there are excess antibiotics, then antibiotics also stop working on them because the bacteria become resistant, which is the third option, and most of you picked that. So all of you who picked the third option, you're correct. But you should remember that the regular function of bacteria dying is also happening. So all of them is the most correct answer here. Okay, TSWREI Society is still leading. And the final question. How can we solve the problem of antibiotic resistance? By using antibiotics as per doctor's prescriptions, by developing new antibiotics, by better diagnostics and healthcare solutions, or all of them. It is all of them, because first one is by using antibiotics as per doctor's prescription, which means that you will not misuse antibiotics. You will use them only when required and you will take the full dose. So bacteria which are going to be exposed to the antibiotics will probably be all cleaned up, all killed. So they cannot reproduce and become resistant ones. So of course you have to use them as per doctor's prescriptions. By developing new antibiotics also, because when bacterial populations become resistant towards one kind of antibiotics, you need other antibiotics to be used in other place. Like Krishna Leela ma'am talked about tuberculosis, right? Where maybe one drug is insufficient, one antibiotic is insufficient, and they need 2 or 3 antibiotics to be used. Now, when those 2 or 3 also become useless against the bacteria, then you need newer antibiotics to be used. So there needs to be research for development of new antibiotics. By better diagnostics, diagnostics help us in finding out what infection you have. For example, in COVID, we use diagnosis for checking if coronavirus is inside our body. Similarly, diagnostics will help us figuring out if you have a bacterial infection or a viral infection. So if we have better diagnostics, then people will know if they have a bacterial infection or a viral infection. And they should be taking antibiotics only when they have a bacterial infection and not viral infection. So we need all of these things to be happening for fighting the problem of antibiotic resistance. So 15 of you who picked all of them, very good. All right, so that is it today, and let's see the final results. All right, Raju has finally caught up again and fetched the first position. Very good, Raju.
Congrats.
All right.
Raju, may I know who is it?
School?
From which school? Can you raise your hand, Raju? Sir, madam. Okay, is it KGM Prasad? K. Mary? Who is Raju? May I know from which school you are, or are you a teacher or a student? Raju Garu, could you please raise hand? Okay. Okay, I don't know. I wanted to talk to him and him or her, I don't know.
Him only,
Yeah.
that is.
Yeah.
Name
Raj.
suggests it is him.
Yeah. Because sometimes the device names could be someone else's as well. So
Yeah.
yeah. But yeah, congratulations. You have even scored more than the immunologist on the panel. Okay. So very good. That means you have listened very attentively to the session.
First question.
Thank you. Please convey the same information to the students in the classroom. What is the problem with overusage of antibiotics? Because it's a really, really burning problem. And very soon we don't have any antibiotics to treat even simple infections.
Okay.
Please remind that. Yeah.
Pavini, there are 3 more questions in the question answer box. So these are the final 3 questions we'll take for the day. Okay.
Sure, sure.
Okay. The question by Lakshmi is there any chance TB attacks twice the same person after cured once, ma'am? So this is possible, but it's very minor. But the possibility rises when you stop taking the medicine as per the doctor's prescription. So generally they prescribe for 9 months. If you take 9 months, most of the time you are not going to get again a second time. But what happens is TB symptoms goes off within 1 week of you are using antibiotics. So people think TB has gone from the body, but that is not correct. Okay, so you should take 9 months. If you have symptoms or not, you should take medicines for 9 months. Then I think the chances of getting second time is very, very less. Until and unless you become very weak and you are infected with some other diseases, your body becomes weak. Otherwise, the chances are very less. Then Bhagya Lakshmi, can bacteria reproduce? Yes, why not? Because they are reproduced, that's why their population is increasing, right? Otherwise, how they will grow in our body? So they reproduce by this binary fission.
So cell division is same as reproduction.
Cell division is same as reproduction. That one cell, you must have learned in your textbooks as a binary division. Asexual reproduction and sexual reproduction, right, in your textbooks. So bacteria divide by asexual reproduction. That is by division. So one cell becomes 2, 2 will become 4. The next is Venkatesh. If we took other antibodies to treat a particular disease, does our body produces against antibodies to control other antibodies? So generally antibodies are produced against an antigen, not against an antibody. Okay. So the foreign material that is coming with the antibodies, maybe our body will react, but not to the antibodies.
No, so outside antibodies, can they behave like antigens for our?
That's what I'm telling. The material that they have used in antibody preparation may trigger the response.
Okay. I think the questions have been fantastic today. Not only there were many questions, I think the quality of questions were also very, very good today. Awesome.
Yeah. Thank you so much.
Thank you all very much. And soon we will meet for the last session of the MIlo CCMB.
Which is on Sundata, may I know the topic?
The topic will be crop improvement. So how plants fight microorganisms and how we can use that to make better crops.
Interesting. Yeah. So thank you so much, everyone, all the teachers. I hope the students are with you and they have attended the session. They've learned more about cell wall, bacteria, antibiotics, and resistance and all that concepts. So we'll see you soon with another session, hopefully before the examination season starts.
Yeah.
Yes.
Sure.
Sundata? Yeah.
Yes.
Thank you, Krishnalila ma'am.
Thank you, ma'am. Thank you for listening.
Bye. Bye.
Bye.